Welcome to the deep dive, where we unpack complex topics and surface with clarity.
Great to be here.
Today, we're diving deep into the unsung heroes of your body,
Oh,
the kidneys.
absolutely. Unsung is the right word.
We all know they filter our blood, right? But what exactly do they do? And I mean, how do they pull off such complex feats every single day to keep your internal environment, perfectly balanced?
It's pretty incredible when you break it down.
Our mission today is to explore Chapter 14 of Vander's Human Physiology, the kidneys and regulation of water and inorganic ions.
Mm.
We'll break down the fundamental principles, intricate structures, and precise control mechanisms that make your kidneys true masters of multitasking.
Yeah, we'll try and make it clear.
Every key term will be explained clearly, making sure you can follow along without needing any visuals, and we'll keep things in the exact order laid out in your textbook.
Right, and what's fascinating here is how this chapter really illustrates homeostasis at its finest. It brings together so many core physiological principles you've probably already encountered. Cellular transport, cardiovascular regulation.
Right, things we've maybe touched on before.
Exactly. For instance, did you know the kidneys can actually make new glucose during prolonged fasting?
Really? I thought that was just the liver.
Most people do. But the kidney stepping in like that, well, it's a critical life-saving backup. A true unsung hero function, like you said.
That's truly astonishing. Okay, let's unpack this then. Beyond that surprising role, what are the primary jobs these remarkable organs handle for us?
Well, the source outlines 5 main functions. First, and maybe most broadly, they're essential for regulating your body's water and inorganic ion balance.
Ions like sodium, potassium.
Exactly. Sodium, chloride, potassium, the big ones. And also your acid-base balance, working, together with the lungs.
Okay, function 1.
Second, they're the primary waste disposal system, removing metabolic byproducts like urea from protein, uric acid from nucleic acids.
Ah, the stuff you don't want building up.
Precisely. And creatinine from muscle creatine, sending them out in your urine.
Got it. Waste removal.
Third, they act as a detox unit, clearing foreign chemicals, think drugs, pesticides, food additives.
So cleaning up external stuff, too.
And their metabolites. Fourth, as you mentioned, that gluconeogenesis, making new glucose when you're fasting for a long time.
The backup energy source.
Right, releasing it into your blood. And finally, number 5, they're like mini hormone factories.
Hormones? What kind?
They produce and secrete erythropoietin that controls red blood cell production.
Okay.
Renin, which is actually an enzyme that regulates blood pressure and sodium balance.
We'll probably come back to that one.
Oh, definitely. And they convert vitamin D to its active form, which influences your calcium balance.
So they're not just filters, they're incredibly precise chemical engineers. That fine-tuning ability sounds absolutely vital.
It really is.
Now let's talk about the architecture.
Yeah.
How are these powerhouses built, and how does urine actually make its way out of the body?
Right. The structure. The overall urinary system is sort of like a plumbing network.
Okay.
Kidneys filter the blood. Urine flows through these muscular tubes called ureters. Then it collects in the bladder.
The storage tank.
Yeah, exactly. And finally exits via the urethra. Your kidneys themselves are tucked away at the back, behind the main abdominal cavity. Retroperitoneal is the term.
Behind the scenes, literally.
Inside, each kidney has an outer layer, the renal cortex, and an inner part, the renal medulla. There's an indented area, the hilum, where blood vessels and the ureter connect.
Like a service entrance?
Sort of, yeah.
Yeah.
Urine drains from collecting areas called calyces into the renal pelvis, which is basically the funnel at the top of the ureter.
Okay, the big picture. But what about the microscopic level?
Ah, that's where the real stars are, the nephrons. Roughly 1 million microscopic functional units in each kidney.
1 million each? Wow.
Yeah. Each nephron has 2 main parts, the renal corpuscle and the renal tubule.
Corpuscle and tubule.
Right. The renal corpuscle is where filtration kicks off. It's basically a compact tuft of tiny, intertwined capillaries called the glomerulus.
Glomerulus. Got it.
It's fed by an afferent arteriole, think arriving, and drained by an efferent arteriole, think exiting.
Afferent in, efferent out.
Exactly. This glomerulus sits inside a cup-shaped thing called Bowman's capsule, creating a space, Bowman's space, where the initial filtered fluid collects, the ultrafiltrate.
Okay, so that first filtering step, you mentioned it's like a high-pressure filter, pushing fluid out.
Yeah, that's a good way to think about it. Blood pressure in the glomerulus is uniquely high compared to other capillaries.
Why is that?
Well, the afferent arteriole is relatively wide, allowing good flow in, And the efferent arteriole offers some resistance on the way out. That builds up the pressure inside the glomerulus.
Makes sense. But what kind of barrier ensures only the right stuff gets through, not blood cells or big proteins?
Good question. The filtration barrier has three layers. There's the capillary wall itself, the endothelium. Then a middle layer, a non-cellular basement membrane. And finally, the inner lining of Bowman's capsule, made of these really unique cells called podocytes.
Podocytes.
Yeah, they have these intricate foot processes that interlock, leaving little filtration slits between them. These layers work together to block cells and almost all proteins.
So the filtrate is basically plasma minus the cells and big proteins.
Exactly. Cell-free and nearly protein-free. From Bowman's capsule, this fluid enters the renal tubule.
The second part of the nephron.
Right. It's a long, winding tube with several distinct segments. First, the proximal tubule.
Proximal meaning close to the start.
Yep. Then it dips down into the hairpin-shaped loop of Henle, with a descending limb going down and an ascending limb coming back up.
Okay. The loop.
Followed by the distal convoluted tubule, distal meaning further away.
Makes sense.
And finally, several nephrons drain into a collecting duct system, which eventually leads to that renal pelvis we mentioned earlier.
So fluid path. Glomerulus, Bowman's capsule, proximal tubule, loop of Henle, distal tubule, collecting duct.
You got it. And the blood supply is special too. Remember the efferent arteriole leaving the glomerulus?
Yeah, the exit one.
It doesn't just go back to the veins. It branches into a second capillary network, the peritubular capillary.
A second set. Why?
These wrap all around the tubules. They deliver oxygen and nutrients to the tubule cells, but crucially, they also pick up all the stuff the tubules reabsorb back from the filtrate.
Ah, so that's how the recycled goods get back into the blood.
Exactly. And about 15% of nephrons, the juxtamedullary ones, meaning near the medulla, have really long loops of Henle that dive deep into the medulla.
Juxtamedullary. Okay. Why the long loops?
They're absolutely key for creating concentrated urine. We'll get into how later.
Okay. Intriguing.
And there's one more structural bit.
Yeah.
The juxtaglomerular apparatus, or JGA.
JGA, what's that?
It's where the ascending limb of the loop of Henle actually loops back and touches the afferent arteriole, feeding its own glomerulus.
Little feedback point.
Precisely. Specialized cells there, the macula densa in the tubule and JG cells in the arterial wall, act as sensors and controllers. They monitor filtrate flow and composition and can influence both filtration rate and blood pressure.
Wow, intricate internal controls. So with this structure in place, how does the kidney actually, clean and recycle the blood? You mentioned three fundamental moves.
Exactly. Three core processes. First is glomerular filtration. That's the initial step. We talked about plasma streaming from the glomerulus into Bowman space. It's like a non-selective first pass, filtering based mostly on size.
Step one, filter almost everything small.
Pretty much. Second is tubular reabsorption. This is the kidney's massive recycling program.
Getting the good stuff back.
Right. Moving vital substances, water, ions like sodium, glucose, amino acids, from the tubular fluid back into the blood in those peritubular capillaries.
Okay. And the third?
Tubular secretion. This is like selective waste removal, the opposite direction of reabsorption. It moves additional waste products, excess ions, or foreign chemicals from the blood into the tubular fluid.
So actively adding things to the to-be-excreted pile.
Exactly. Ensuring they get eliminated. Plus, the tubule cells themselves can metabolize some substances.
Okay, so filter, reabsorbs, secrete.
Yep. The kidney's work boils down to a simple equation. What you ultimately excrete, what got filtered, plus what got secreted, what got reabsorbed.
Amount out filtered plus secreted reabsorbed. Makes sense.
But remember, not every substance undergoes all three. Some are only filtered and reabsorbed, some filtered and secreted, some just filtered.
Right. It depends on the substance. Can you give an example?
Sure. Think of, say, a toxin or certain drug metabolites. They get filtered and they get actively secreted, but they're not reabsorbed. So the kidney gets rid of them very efficiently. Maximum excretion.
Okay, clear it out fast.
Now contrast that with glucose, vital energy source. It gets freely filtered, but then under normal conditions, it gets completely reabsorbed back into the blood in the proximal tubule.
So none ends up in the urine.
Normally zero. Your body holds on to all of it. The kidney manages each substance specifically based on the body's needs.
That's incredibly selective. Let's zoom in on that first step again, glomerular filtration. What are the forces involved?
It's driven by Starling forces, similar to other capillaries, but the balance is different here. The main driving force is that high hydrostatic pressure inside the glomerular capillaries, the blood pressure, PGC. It's about 60 millimere G, much higher than typical capillaries.
60, okay. Pushing fluid out.
Right. Opposing that push are two forces. First, the hydrostatic pressure of the fluid already in Bowman space,
Hmm.
PBS, pushing back. That's about 15 millimeter Hg.
Okay, resisting force 1.
And second, the osmotic force, due to the proteins trapped in the glomerular capillary plasma, pi GC. They pull water back in. That's around 29 millimeter Hg.
Resisting force 2, proteins pulling water back.
Exactly. So the net glomerular filtration pressure is PGC minus PBS minus pi GC. So 60 minus 15 minus 29 equals 16 millimeters G.
16. Still a positive pressure pushing fluid out?
Always positive under normal conditions, yeah. That drives filtration. And the volume of fluid filtered per unit time is the glomerular filtration rate, or GFR.
GFR, you said that was huge.
Staggering. It averages about 180 liters per day, or 125 milliliters per minute.
180 liters.
Yeah.
Well, my whole plasma volume is maybe 3 liters.
Exactly.
Yeah.
Your kidneys filter your entire plasma volume roughly 60 times every single day.
Wow, 60 times a day. That continuous processing must be why they can keep things so stable.
Precisely. It allows for very fine adjustments on each pass. And GSR isn't fixed. It's tightly regulated.
How so?
Nerves and hormones can signal the smooth muscles in those afferent and efferent arterioles to constrict or dilate.
Changing the pressure inside the glomerulus.
You got it. For example, if the afferent arteriole constricts, less blood flows in, PGC drops, and GFR decreases.
Okay. And if the efferent constrict?
Then blood backs up a bit in the glomerulus, PGC increases, and GFR goes up. It's a delicate balance.
Makes sense. Control knobs for filtration.
Right. And there's another term related to GFR, filtered load. That's simply the GFR multiplied by the plasma concentration of a given substance. It tells you how much of that substance enters the tubules per minute.
Filtered load. GFRX, plasma concentration.
Yep.
Useful for comparing things.
Very useful for figuring out how much reabsorption or secretion is happening later on. Okay, so after filtration, we get to tubular reabsorption.
Getting the good stuff back.
Exactly. Remember those huge filtered loads? 180 liters of water, tons of sodium, glucose. Most of this useful stuff gets almost completely reabsorbed, over 99% for water and sodium.
99%, that's efficient.
Hugely efficient. Waste products like urea are reabsorbed too, but much less completely, allowing them to be excreted.
So how does this reabsorption happen? Is it just flowing back?
No, it's not bulk flow like filtration. It's specific transport mechanisms. Either diffusion, moving passively down concentration gradients,
Okay.
or more often, mediated transport, which requires specific protein carriers or channels in the tubule cell membranes.
Protein helpers.
Yep. It's often transcellular transport across the apical membrane, facing the tubulumen, through the cell cytoplasm, and then across the basolateral membrane, facing the interstitial fluid and blood.
Across one side, through the middle, out the other.
Exactly. Take sodium, for example. It typically enters the cell passively from the lumen down its gradient. But getting it out of the cell on the basolateral side requires active transport.
The sodium pump.
The NABI plus K plus ATPase pump. It uses energy to pump sodium out into the interstitial fluid, keeping the intracellular sodium concentration low, which maintains the gradient for sodium to enter from the lumen.
The pump is key for keeping the whole process going.
Absolutely central.
Yeah.
And many other substances hitch a ride with sodium. Glucose, amino acids, some ions that use secondary active transport, co-transported with sodium as it moves down its gradient into the cell.
Using sodium's downhill movement to pull other things uphill. Clever.
Very clever. But these mediated transport systems, the ones using proteins, have a limit.
A speed limit.
Sort of. It's called the transport maximum, or TEM. There are only so many binding sites on the transport proteins. If the concentration of the substance in the filtrate is too high, the transporters get saturated.
You can't keep up.
Right. Any amount filtered beyond the TEM can't be reabsorbed and ends up being excreted in the urine.
Like the glucose example in diabetes.
Exactly. Normally, blood glucose is low enough that the filtered load is well below the TEM for glucose reabsorption. So all glucose is reabsorbed.
Right.
But in uncontrolled diabetes, blood glucose skyrockets. The filtered load of glucose exceeds the TEM. The transporters are overwhelmed, and glucose spills into the urine. That's called glucosuria.
Okay, Tim explains that. Now, what about secretion? Adding stuff to the urine.
Right, tubular secretion. Moving substances from the peritubular capillary blood into the tubular lumen, like reabsorption and reverse.
Also,
Yes, can be diffusion or mediated transport. It's really important for getting rid of excess hydrogen ions, H+, to control pH, excess potassium ions, K+, and many organic anions, including lots of drug metabolites and things like penicillin.
So it fine-tunes H plus and K plus A and helps clear certain wastes and drugs faster.
Exactly. Faster than filtration alone could. Now, different parts of the tubule specialize. There's a division of labor.
Okay, who does what where?
The proximal tubule is the workhorse. It does the bulk reabsorption of most filtered solutes, and water like 2/3 of it. It's also the main site for secreting many organic substances, but not potassium.
Proximal tubule bulk processing.
Pretty much. Then the loop of Henle, especially those long juxtamedullary ones, is crucial for setting up the kidney's ability to concentrate urine. It reabsorbs a good chunk of salt and some water.
Loop of Henle concentration power.
Right. And finally, the later segments, the distal convoluted tubule and the collecting ducts, that's where the real fine-tuning happens for most substances.
The final adjustments.
Exactly. This is where most of the hormonal control happens, determining the final amounts excreted based on the body's moment-to-moment needs. Adjusting sodium, potassium, water, calcium. It all happens here.
Okay, that division makes sense. Bulk work first, then specialization, then fine-tuning.
You got it. Now, how do we actually measure how well the kidneys are doing all this? That brings us to renal clearance.
Right, you mentioned this earlier, measuring kidney function.
Yes. Renal clearance is a really important concept. It's defined as the volume of plasma from which a substance is completely removed or cleared by the kidneys per unit of time.
Volume of plasma cleared per minute, usually.
Yep, typically ml/min. The formula is like this. Clearance of substance S, we write it CS, equals the urine concentration of S, US, times the urine volume per time, V, all divided by the plasma concentration of S.
PS. Okay, CUXVP.
Exactly. Let's think about some examples. Glucose. Normally, what's its urine concentration?
0, you said, because it's all reabsorbed.
Right, so glucose is 0. That means the glucose clearance is normally 0. Makes sense, no plasma is being cleared of glucose.
Okay, but if glucose does show up in urine, like in diabetes,
then glucose becomes positive and the clearance becomes positive. It tells you the T's was exceeded or there's some renal disease affecting reabsorption.
I see. What about using clearance to measure GFR?
Yes. For that, you need a substance that's freely filtered but not reabsorbed and not secreted.
Just filtered.
Right. If that's the case, then the amount filtered per minute, GFR XPS, must equal the amount excreted per minute, USXV.
Okay, so GFR-XP equals USV.
Exactly. Rearrange that and you get GFR USXV PS, which is the clearance formula.
So the clearance of such a substance is the GFR.
Precisely. The substance used experimentally for this is inulin, a polysaccharide. It fits the criteria perfectly.
Inulin clearance useful for research.
Very. But giving someone inulin isn't practical clinically, so we use something the body already produces. Creatinine.
Creatinine clearance.
Yes. Creatinine is a waste product from muscle metabolism. It's freely filtered and not reabsorbed. It is secreted a tiny bit, though.
So not perfect like inulin.
Not absolutely perfect. Because of that small secretion, creatinine clearance slightly overestimates the true GFR.
But close enough for clinical use.
Yes, it's a very useful and common clinical estimate of GFR. If your blood creatinine level goes up, it often signals that your GFR has decreased. Your kidneys aren't clearing it as effectively.
Okay, that makes sense. What about clearance telling us about secretion or reabsorption?
Right. Compare the substance's clearance to the GFR, measured by inulin or estimated by creatinine clearance.
Okay.
If the substance's clearance is greater than GFR, what must be happening?
More is ending up in the urine than just got filtered, so it must be secreted too.
Exactly. Net secretion is occurring. Conversely, if a substance is filterable, but its clearance is less than GFR,
then less is in the urine than was filtered, so it must be reabsorbed.
Precisely. Net reabsorption is occurring.
So clearance gives us a window into these processes. What's the key takeaway here for, say, understanding medication?
It's crucial. If a doctor knows a drug's clearance is much higher than the patient's GFR, it tells them the kidneys are actively secreting that drug. They're working hard to eliminate it.
Which might affect how often you need to take it.
Exactly. It impacts dosing strategies. Knowing how the kidney handles a drug filtration, reabsorption, secretion is fundamental pharmacology.
Got it. All right, so after all this complex processing, filtration, reabsorption, secretion, the final product is urine. How does the body actually, get rid of it? The micturition part.
Right, micturition or urination. Urine flows from the renal pelvis down the ureters. This isn't just gravity. The ureter walls have smooth muscle that contracts rhythmically to propel the urine.
Pushing it along.
Yep. It gets stored in the bladder, which is essentially a muscular bag lined with smooth muscle called the detrusor muscle.
Detrusor, that's the muscle that contracts to empty the bladder.
Correct. At the bladder's exit, where it joins the urethra, there are two sphincters, like valves. An internal urethral sphincter, made of smooth muscle.
Involuntary.
Right. It's normally passively closed when the detrusors are relaxed. And below that, an external urethral sphincter, made of skeletal muscle.
Skeletal muscle, so
Right.
voluntary control.
Exactly. You control that one consciously.
So how does the reflex work? When you gotta go, you gotta go.
Well, as the bladder fills with urine, the walls stretch. Stretch receptors in the bladder wall send signals via afferent nerves to the spinal cord.
Okay, signal in.
This triggers a spinal reflex. Parasympathetic nerves fire, causing the detrusor muscle to contract.
Squeezing the bladder.
Yep. That contraction pulls the internal sphincter open. At the same time, the signals inhibit the sympathetic nerves that help keep the internal sphincter closed. And, crucially, they inhibit the somatic motor neurons going to the external sphincter.
So relax the external sphincter too.
Right. Detrusor contracts, both sphincters open, and urination occurs. That's the basic involuntary reflex.
Well, we can hold it.
We can. Descending pathways from the brain allow for voluntary control over this reflex. You can consciously keep the external sphincter contracted, even when the detrusor starts contracting.
Ah, overriding the reflex.
Yes. And you can also initiate urination voluntarily by relaxing the external sphincter and often consciously contracting abdominal muscles to increase bladder pressure. This is learned during toilet training.
Fascinating interplay of reflex and voluntary control. Sometimes that control fails though, right? Incontinence.
Yes, incontinence is involuntary urine release. It's quite common, especially in older women. There's stress incontinence, like leakage during coughing or exercise, often due to weakened pelvic support,
Okay.
and urge incontinence or overactive bladder where the detrusor contracts unexpectedly. This might be due to neurological issues or bladder irritation, often treated with drugs that block the parasympathetic signals to the detrusor.
Okay. Let's pivot now to the bigger picture, regulating the amount of water and salt in the whole body.
Right, total body balance. Water is huge, making up 55–60% of your body weight.
Mostly water.
Yep. We gain water from liquids, food, and even metabolism. We lose it through skin and lungs, insensible loss, plus sweat, feces, and urine.
And the kidneys control the urine part.
They're the major control point for water loss. They can adjust urine output dramatically, from less than half a liter a day if you're dehydrated, up to maybe 25 liters if you drink excessive water.
25 liters. Incredible range. What about salt? Sodium chloride.
Similar story. We gain salt mainly from food. We lose small amounts in sweat and feces, but the vast majority of regulated loss is via urine. Kidneys can adjust NaCl excretion over a very wide range to precisely match your intake or other losses.
Maintaining that balance. So how do they handle sodium and water at the nephron level?
Well, both sodium and water are freely filtered to the glomerulus. Then, they both undergo massive reabsorption. Over 99% usually gets reabsorbed back into the blood.
Okay, huge reabsorption. Is there any secretion?
Nope. No secretion for sodium or water. So excretion is purely controlled by regulating filtration and, much more importantly, reabsorption.
Mostly about controlling reabsorption then.
Definitely. Two key generalizations here. One, sodium reabsorption is an active process requiring energy in almost all parts of the tubule except the descending limb of the loop of Henle.
Active sodium pumping?
Two, water reabsorption is passive, driven by osmosis. It follows the solutes, especially sodium.
Water follows salt, the old saying.
Exactly. Sodium gets pumped out of the tubule fluid into the interstitial space. This makes the interstitium saltier and the tubule fluid less salty, creating an osmotic gradient. Water then moves passively down this gradient out of the tubule and into the interstitium, eventually entering the blood.
So sodium movement creates the gradient for water movement.
Precisely. The primary driver is at Ni plus K plus ATPase pump on the baselateral membrane of the tubule cells, pumping sodium out. This keeps cells sodium low, allowing sodium to enter from the lumen passively through various channels or co-transporters depending on the tubule segment.
Like the glucose co-transporter we mentioned.
Right, or sodium-hydrogen exchangers, or specific sodium channels in later segments. But the key is that active pumping out the basolateral side drives the whole thing.
Okay. But for water to follow, the tubule has to be permeable to water, right?
Absolutely critical point. Water permeability relies on water channels called aquaporins in the cell membranes.
Aquaporins, water pores.
Yep. In the proximal tubule, there are always lots of aquaporins. So as sodium is reabsorbed, water automatically follows in proportion. About 2/3 of sodium and water reabsorption happens here, obligatorily linked.
Okay. Always permeable in the proximal part.
But in the later parts, specifically the collecting ducts, the water permeability is variable and physiologically controlled.
Ah, this is where the fine-tuning comes in.
Yes, and the main controller of this variable water permeability is the hormone vasopressin.
Vasopressin, also called ADH, antidiuretic hormone.
Correct. ADH. It's released from the posterior pituitary gland in your brain. When vasopressin levels are high, it acts on the collecting duct cells.
What does it do to them?
It triggers a signaling cascade involving KMP that causes vesicles containing AQP2 aquaporin channels to fuse with the apical membrane, the one facing the tubular fluid.
So it inserts water channels into the membrane.
Exactly. Makes the membrane highly permeable to water. Water then rushes out of the collecting duct following the osmotic gradient into the salty interstitial fluid of the medulla and gets reabsorbed into the blood.
Resulting in less urine.
Much less urine, and the urine becomes very concentrated, maximum water conservation.
And if vasopressin is low?
Then those AQP2 channels are removed from the apical membrane. The collecting duct becomes relatively impermeable to water. Water can't leave easily, so it stays in the tubule.
Resulting in?
A large volume of dilute urine. This is called water diuresis, getting rid of excess water.
So vasopressin is like the tap controlling water reabsorption in the collecting ducts.
Perfect analogy. High vasopressin, tap is wide open, lots of reabsorption. Low vasopressin, tap is closed, water stays in the pipe and flows out.
What happens if that system breaks? You mentioned diabetes insipidus.
Right. That's caused either by failure to produce or release vasopressin from the brain's central DI, or the kidneys failing to respond to vasopressin nephrogenic DI.
Either way, no vasopressin effect.
Right. So the collecting ducts stay impermeable to water. The person produces huge volumes, up to 25 liters a day, of very dilute urine. Constant water loss, leading to severe dehydration and high plasma osmolality if they can't drink enough to keep up.
Wow. That highlights how crucial vasopressin is. Is that different from the diuresis in regular diabetes mellitus?
Yes, good distinction. In uncontrolled diabetes mellitus, you have excess glucose in the filtrate, osmotic agent. This glucose holds onto water in the tubule by osmosis, preventing its reabsorption even if vasopressin is present. That's called osmotic diuresis, increased urine flow due to excess solutes.
So osmotic diuresis is solute-driven water loss, while water diuresis, low vasopressin, is just water loss without necessarily losing extra solute.
Exactly. Key difference. Now, how does the kidney actually generate that really salty environment in the medulla that allows vasopressin to concentrate the urine so effectively?
Yeah, you said it could get up to 1,400 mosmol while blood is only around 300. How is that possible?
It's the magic of the countercurrent multiplier system, mainly involving the loops of Henle of those juxtamedullary nephrons.
Countercurrent multiplier. Sounds complex.
It is clever. Think about the loop of Henle. Fluid flows down the descending limb and then up the ascending limb opposite directions. That's the countercurrent part.
Okay, down then up.
Now the key properties. The ascending limb actively pumps out salt, NaCO, into the surrounding interstitial fluid. But, and this is critical, the ascending limb is impermeable to water.
Pumps salt out, but water can't follow.
Right, so the interstitial fluid gets saltier and saltier. Now consider the descending limb, which is permeable to water, but doesn't pump salt.
Okay, water can move there.
As fluid flows down the descending limb, it enters the increasingly salty environment created by the ascending limb. So water passively diffuses out of the descending limb into the salty interstitium.
Making the fluid inside the descending limb more concentrated as it goes down?
Exactly. It equilibrates with the surrounding salty fluid. So by the time the fluid reaches the bottom of the loop deep in the medulla, it's very concentrated, maybe 1,200, 1,400 mosmol.
Wow.
Then this concentrated fluid turns the corner and enters the ascending limb. What does the ascending limb do?
Pumps salt out, but is impermeable to water.
Right. So as it pumps salt out, the fluid inside the ascending limb becomes progressively more dilute as it travels upwards. By the time it leaves the loop and enters the distal tubule, it's actually hypoosmotic, maybe 100 mosmol.
Dilute again. So the loop itself makes the medulla salty, but leaves the tubular fluid dilute.
Precisely. It multiplies the concentration gradient in the medulla. The continuous flow and pumping establishes this standing gradient, very salty deep inside, less salty near the cortex.
Okay, I think I get the countercurrent multiplier idea. So the fluid entering the collecting duct is dilute.
Yes, around 100 mosmol. Now what happens next depends entirely on vasopressin.
Ah, the tap control again.
No, exactly. If vasopressin is high, the collecting duct becomes permeable to water. as this dilute fluid flows down through the increasingly salty medulla.
Water gets sucked out by osmosis into that salty interstitium.
Bingo. Leaving the remaining fluid inside the collecting duct to become progressively more concentrated, reaching maybe 1,400 mosmol by the end, small volume, highly concentrated urine.
And if vasopressin is low,
Collecting duct stays impermeable. The dilute fluid just flows right through, maybe getting slightly more dilute, and you excrete a large volume of hypoosmotic urine.
amazing system. What about the blood vessels down there, the vasa recta? Don't they wash away the salt gradient?
Good question. They don't, because they also form hairpin loops that run parallel to the loops of Henle, a countercurrent exchange system.
Countercurrent again?
Yes. As blood flows down into the salty medulla, salt diffuses in and water diffuses out. But as the blood loops back towards the cortex, moving through less salty areas, the opposite happens. Salt diffuses out and water diffuses back in.
So it picks up salt on the way down and drops it off on the way up, minimizing washout.
Exactly. It helps maintain the medullary gradient. And one final player in that gradient is urea.
Urea, the waste product.
Yep. Vasopressin actually increases the permeability of the inner medullary collecting duct to urea. Urea then diffuses out into the deep medullary interstitium, adding significantly to its osmolality, especially when you're trying to conserve water, high vasopressin state. Some of this urea gets recycled back into the loop of Henle, trapping it in the medulla.
So urea recycling helps make the medulla even saltier.
It contributes substantially, yeah. So it's salt pumping by the ascending limb, plus urea trapping in the inner medulla that creates that super high osmolarity needed to concentrate urine.
Okay, that covers water balance and concentration. What about regulating sodium itself? That seems crucial for blood volume and pressure.
Absolutely central. Remember, sodium excretion is the difference between what's filtered and what's reabsorbed. The kidneys control both GFR and, much more significantly, the rate of tubular sodium reabsorption.
Controlling reabsorption is the main game.
It is. Interestingly, your body doesn't have specific receptors that directly monitor your total body sodium level.
No sodium ostat.
Not really. Instead, regulation is mainly initiated indirectly through changes in blood volume and pressure, which are sensed by baroreceptors in the cardiovascular system.
Ah, so low sodium leads to low volume pressure, which triggers a response.
Exactly. Low total body sodium means lower plasma volume, which means lower blood pressure. Baroreceptors detect this and trigger reflexes.
What do the reflexes do to the kidneys?
Two main things. They cause a decrease in GFR, filtering less sodium to begin with.
Okay, filter less.
And, more importantly, they trigger mechanisms that increase tubular sodium reabsorption.
Get more back.
Filter less, reabsorb more. Result is decreased sodium excretion.
Precisely. Helping the body hold on to sodium, which also helps hold on to water, restoring volume and pressure.
How is GFR decreased?
Mostly through sympathetic nerve activity constricting the afferent arterioles. Lower arterial pressure itself also contributes. And how is sodium reabsorption increased? This sounds like hormones might be involved.
Bingo. The major hormonal controller of sodium reabsorption is aldosterone.
Aldosterone. From the adrenal glands.
Yes, from the adrenal cortex, sits on top of the kidneys. Aldosterone acts mainly on the distal convoluted tubule and the cortical collecting ducts.
What does it do there?
It stimulates those cells to reabsorb more sodium. It basically tells them to make and insert more sodium channels on the apical side and more Na plus K plus ATPase pumps on the basolateral side. More pathways for sodium to get back into the blood.
Turn up the sodium pumps and channels.
Essentially, yes. Now, what controls aldosterone secretion? That's where the renin-angiotensin system, or RAS, comes in, often called RAAS, for renin-angiotensin-aldosterone system.
Okay, the RAS connection, how does it work?
It starts with renin, remember renin, the enzyme released by those juxtaglomerular JG cells in the kidney.
Yeah, from the JGA. When is it released?
It's released in response to signals that indicate low sodium or low blood pressure. Three main signals.
Okay, what are they?
1. Increased activity of renal sympathetic nerves, triggered by the cardiovascular baroreceptors, sensing low pressure.
Nerves tell JG cells to release renin.
2. The JG cells themselves act as intrarenal baroreceptors. If pressure in the afferent arteriole drops, they directly sense it and release more renin.
Kidney's own pressure sensor.
3. The macula densa cells in the distal tubule sense the composition of the fluid flowing past. If they detect decreased sodium delivery, implying low GFR or low plasma sodium, they send paracrine signals to the nearby JG cells to release renin.
So nerves direct pressure sensing, and filtrate sensing all converge on renin release. That's quite a complex feedback loop, isn't it?
It is. Multiple layers of control, all aimed at activating renin release when sodium or pressure is low.
So renin is released, what then?
Renin acts on a plasma protein called angiotensinogen, made by the liver, always circulating. Renin cleaves it into angiotensin first.
Angiotensin the first, is that active?
Nope, inactive. It then gets converted into the active form, angiotensin II, by another enzyme called angiotensin-converting enzyme, or ACE.
ACE. I've heard of ACE inhibitors for blood pressure.
Exactly. ACE is found mainly in the capillaries of the lungs. Angiotensin II is the real powerhouse.
What does angiotensin II do?
Two major things relevant here. One, it stimulates the adrenal cortex to secrete aldosterone.
Closing the loop to increase sodium reabsorption.
Right. Two, angiotensin II is a potent vasoconstrictor itself. It constricts arterioles throughout the body, directly increasing blood pressure.
Yeah. So it tackles both sodium retention and blood pressure directly.
Yes. It's a powerful system for responding to low volume or low pressure. And those drugs, ACE inhibitors like lisinopril or angiotensin II receptor blockers, ARBs like losartan, or even aldosterone blockers, they all target this system to lower blood pressure in people with hypertension.
Makes sense why they're so effective. Is there anything that counters this system? What if blood volume is too high?
Yes, there is. Atrial natriuretic peptide, or AMP.
AMP, from the heart.
Yes, secreted by cells in the atria of the heart when they get stretched too much, which happens when blood volume is high.
So the heart senses high volume and releases ANP. What is ANP?
It does pretty much the opposite of the RAAS. It inhibits sodium reabsorption in the tubules, it inhibits aldosterone secretion, and it can even increase GFR slightly.
All leading to more sodium excretion.
Exactly. More sodium and therefore more water excretion, natriuresis, and diuresis helps bring blood volume back down.
So RAAS retains sodium when low, ANP excretes sodium when high, a balance.
A beautiful balance. And there's one more factor. Pressure natriuresis. Just high blood pressure itself seems to directly inhibit sodium reabsorption in the tubules, causing more sodium loss. It's another link between pressure and volume.
Okay, that covers sodium regulation. What about water regulation specifically? We know vasopressin controls permeability, but what controls vasopressin release?
Right. Water excretion depends on vasopressin. Two main inputs control vasopressin secretion from the posterior pituitary. The most important one under normal conditions is plasma osmolarity.
Osmolarity, the concentration of solutes in the blood.
Exactly. Specialized neurons in the hypothalamus called osmoreceptors constantly monitor blood osmolarity.
Okay, what happens if osmolarity goes up, like when you're dehydrated?
The osmoreceptors are stimulated. They signal the vasopressin-releasing neurons to fire more, releasing more vasopressin into the blood.
More vasopressin leads to
More water reabsorption in the collecting ducts. Small volume concentrated urine conserves water, helps bring osmolarity back down towards normal.
And if osmolarity goes down, like after drinking lots of water,
Osmoreceptors are inhibited. Vasopressin release decreases sharply. Collecting ducts become impermeable to water.
leading to
Large volume, dilute urine, excretes the excess water, brings osmolarity back up towards normal.
So osmoreceptors provide very sensitive control of water balance to maintain osmolarity. You mentioned the separation of water and sodium excretion enabled by vasopressin is key.
Absolutely key. It allows your body to regulate total fluid volume, mainly via sodium control with aldosterone, and fluid concentration via water control with vasopressin, somewhat independently. You can adjust one without necessarily messing up the other too much.
Clever. What's the second input to vasopressin?
Baroreceptors, the same ones involved in RAS control.
Sensing blood pressure or volume.
Exactly.
Yeah.
If there's a significant decrease in plasma volume or blood pressure, like during severe dehydration or hemorrhage, the baroreceptors fire less, and this signals increased vasopressin release.
More vasopressin to retain water.
Yes, it's another mechanism to help defend blood volume in emergencies. Vasopressin also causes vasoconstriction at these high levels, helping to support blood pressure, hence its name, vasopressin.
So both osmoreceptors and baroreceptors control vasopressin, but maybe on different scales.
Right. The osmoreceptor control is very sensitive to small changes in osmolarity, operating constantly. The baroreceptor input requires a larger change in volume or pressure, maybe a 10–15% drop, so it's more important in more severe situations.
Got it. Osmolarity is the fine-tuner. Baroreceptors are the emergency backup for volume.
Good way to put it. And other things like pain, stress, certain drugs can also influence vasopressin release via inputs from other brain areas.
Okay, let's tie this together. How do all these systems, RAAS, AMP, vasopressin work together in a real-world scenario, like when you're sweating buckets during intense exercise.
Great example. Severe sweating causes loss of hypoosmotic fluid. You lose more water than salt, relatively speaking.
So two problems: decreased extracellular volume and increased body fluid osmolarity.
Exactly. So what happens? The decreased plasma volume is sensed by baroreceptors.
Triggering.
Increased sympathetic output, decreased stretch in the afferent arterioles, probably decreased sodium delivery to the macula densa, all stimulating renin release.
Activating the RAAS.
Right. Increased renin leads to increased angiotensin II, which leads to increased aldosterone.
And aldosterone increases sodium reabsorption.
Check. Meanwhile, the increased plasma osmolarity is sensed by the hypothalamic osmoreceptors.
Diagram.
Increased vasopressin secretion.
Leading to increased water reabsorption.
Check. Plus, GFR likely decreases a bit due to sympathetic constriction and lower pressure.
So the net result is?
Kidneys work hard to retain both sodium, thanks to aldosterone, and water, thanks to vasopressin, minimizing further losses, hoping to stabilize volume and prevent osmolarity from rising too much further. A beautifully integrated response.
It really is. But the kidneys can only conserve what's already there, right? They can't create new water or salt. So how do we replace losses?
Excellent point. That's where behavior comes in. Thirst and salt appetite.
Thirst. What triggers that?
Primarily the same two stimuli that trigger vasopressin release. Increased plasma osmolarity, sensed by hypothalamic osmoreceptors, this is the most important stimulus normally, and decreased extracellular fluid volume, sensed by baroreceptors. Angiotensin II might also play a role in stimulating thirst.
So the same signals that tell the kidney to save water also tell the brain, go drink water.
Makes perfect sense, doesn't it?
Yeah.
Plus other factors like dryness of the mouth contribute.
And salt appetite. Do we crave salt when we're low?
Humans definitely have a strong hedonistic appetite for salt. We just like the taste, often leading to intake far above our needs. True regulatory salt appetite, where you specifically seek out salt because you're deficient, is relatively weak in humans compared to some animals, unless the deficiency is really severe.
Interesting. Okay, let's quickly cover potassium regulation. Why is that important?
Potassium K+ is the main intracellular ion, but the concentration outside the cells in the extracellular fluid is critical for the function of excitable tissues, nerves, and muscles, especially the heart.
Because it affects the resting membrane potential.
Exactly. Too high extracellular K+, hyperkalemia, or too low, hypokalemia, can cause serious problems like cardiac arrhythmias or muscle weakness.
So keeping it in a narrow range is vital. How do kidneys manage it?
Renal regulation is the major control. Potassium is freely filtered. Most of it gets reabsorbed, mainly in the proximal tubule and loop of Henle, but the key control point is potassium secretion in the cortical collecting ducts.
Secretion again, adding K+ to the urine there.
Yes. Changes in potassium excretion are mainly due to changes in how much is secreted by this segment.
How does that secretion work?
It relies on the Na plus K plus ATPase pump on the basolateral membrane. It pumps K+ into the cell from the blood. This raises intracellular K+ OCHO, creating a gradient for K+ to diffuse out of the cell through K+ channels in the apical membrane into the tubular lumen.
So the pump brings it in, channels let it out into the urine. What controls this?
Two main factors directly influence K+ secretion here. First, the plasma K+ concentration itself. If plasma K+ goes up, it directly stimulates the Na plus K plus ATPase pump activity, leading to more K+ uptake into the cell and thus more secretion.
Higher plasma K+ directly leads to more K+ excretion. Simple feedback.
Yep. Second factor, aldosterone.
Aldosterone again, didn't that reabsorb sodium?
It does.
Yeah.
And, conveniently, it simultaneously enhances potassium secretion in the same cortical collecting duct cells. It stimulates both the Na plus K+ pump and the apical K+ channels.
So aldosterone saves sodium but gets rid of potassium.
It's exactly a 2-for-1 deal. And interestingly, the adrenal cortex cells that secrete aldosterone are also directly sensitive to plasma K+ concentration. High plasma K+ directly stimulates aldosterone release, which then promotes K+ secretion by the kidney.
Wow, so high K+ triggers its own excretion via two routes, direct effect on kidney cells and indirectly via aldosterone.
Right. Ensures tight control.
Okay, quick look at calcium and phosphate.
Sure. Their balance is mainly controlled by parathyroid hormone, PTH, and active vitamin D, 1025-OH2D. Calcium is filtered, most reabsorbed proximally without hormone control. The fine-tuning happens later in the distal tubule and collecting duct, regulated by PTH.
How does PTH affect calcium?
If plasma calcium is low, PTH secretion increases. PTH then stimulates calcium reabsorption in these distal segments, helping to bring plasma calcium back up. PTH also stimulates the activation of vitamin D by the kidneys.
Okay. And phosphate.
Phosphate is also filtered and mostly reabsorbed proximally. But PTH has the opposite effect on phosphate compared to calcium. PTH inhibits phosphate reabsorption.
So PTH saves calcium but makes you excrete phosphate.
Correct. Helps raise plasma calcium without raising phosphate simultaneously.
Okay, before we wrap up, can we briefly touch on diuretics? How do they fit into this?
Good question. Diuretics are drugs designed to increase urine volume, primarily by increasing salt and water excretion. Most work by inhibiting sodium reabsorption at different points along the nephron.
If you block sodium reabsorption, water stays with it.
Exactly. Water follows salt. Different classes target different transporters. For example, loop diuretics, like furosemide, Lasix, are very powerful. They block the NADK2Cl co-transporter in the ascending limb of the loop of Henle.
Blocking that key salt pump in the loop.
Right. Leads to significant loss of sodium and water. A common side effect can be potassium loss, too.
Okay. Are there diuretics that don't waste potassium?
Yes, potassium-sparing diuretics. They work later in the cortical collecting duct. Some block the action of aldosterone, like spironolactone. Others directly block the apical sodium channels there. They inhibit sodium reabsorption without increasing potassium secretion.
Useful if potassium levels are a concern.
Definitely. There are also osmotic diuretics, like mannitol. They are filtered but not reabsorbed, so they stay in the tubule and hold water there by osmosis, increasing urine flow.
Like the glucose effect in diabetes mellitus?
Similar principle, yes. Diuretics are widely used to treat conditions involving salt and water retention, like edema and heart failure, and also to treat hypertension by reducing blood volume.
Makes sense. Okay, we've covered a massive amount of ground on kidney function and regulation.
We really have. From basic filtration to complex hormonal control.
Let's try to quickly recap the main takeaways from this deep dive into Chapter 14.
Sounds good.
So we started with the kidney's 5 key functions: regulating water and ions, removing waste, clearing foreign chemicals, making glucose and fasting, and producing crucial hormones like EPO and renin, plus activating vitamin D.
Right, a multitasking powerhouse.
Then we looked at the structure, the overall urinary system, the kidney's cortex and medulla, and the million nephrons per kidney, each with a renal corpuscle, glomerulus, and Bowman's capsule for filtration.
And a long renal tubule, proximal, loop of Henle distal collecting duct for reabsorption and secretion. Plus that unique 2-capillary system, glomerular and peritubular.
We unpack the 3 basic renal processes: glomerular filtration, driven by pressure, creating about 180 Ls of filtrate daily.
Tubular reabsorption, getting almost all the useful stuff back via diffusion and mediated transport, often limited by a TM.
And tubular secretion, actively adding wastes and excess ions to the filtrate. The final amount excreted, ilk filtered plus secreted reabsorbed. We learned about GFR and how renal clearance, CUVP, helps measure it using inulin or creatinine, and tells us if a substance is reabsorbed, CGFR, or secreted CGFR.
Crucial concepts for understanding function and drug handling.
We covered micturition, the bladder, sphincters, and the mix of reflex and voluntary control.
Yep.
Then the huge topic of regulation. Water balance depends on vasopressin ADH, controlling aquaporin insertion and collecting ducts, allowing concentration via the countercurrent multiplier system in the medulla, driven by salt pumping in the loop and urea trapping.
Vasopressin itself is controlled mainly by osmoreceptors and, in emergencies, baroreceptors.
Sodium balance relies heavily on controlling reabsorption via the renin-angiotensin-aldosterone system, RAAS, activated by low-pressure volume sodium, which increases aldosterone to save sodium.
Counteracted by ANP from the heart when volume is high.
Potassium balance involves controlled secretion in the cortical collecting ducts, stimulated by high plasma K+ and by aldosterone.
And calcium phosphate balance involves PTH, which saves calcium but promotes phosphate excretion.
Phew, that's a lot. It really highlights that division of labor bulk processing early on, fine-tuning later under hormonal control.
Absolutely. If we connect this to the bigger picture, these seemingly small organs are just incredible regulators. They aren't just filters. They're sophisticated chemical plants constantly adjusting dozens of variables with amazing precision.
Truly masters of homeostasis.
It makes you wonder, next time you take a sip of water or maybe feel that urge to use the restroom after studying hard, just consider the silent, tireless work going on inside.
Constant adjusting, filtering, balancing.
Yeah, just keeping every single cell in your body happy and functioning in that perfectly balanced internal sea. It's really quite something when you think about how complex yet resilient the system is.
It really is. And with that thought, we've reached the end of our deep dive into renal physiology based on Vander's Chapter 14. Thank you so much for joining us on this learning journey.
Thanks for listening.
From the entire Last Minute Lecture team, thank you for tuning in.